All right, so welcome to lecture number 2 of Biochemistry and Society, which is atoms, molecules, and life. So this is a really interesting lecture, I think. We're basically going to start from the complete formation of the universe and the formation of atoms and start from there. So by the end of this lecture, you should have a really good understanding of where all the atoms in biochemistry come from, all the atoms in the universe come from, basically. Another important part is we're going to talk about how atoms work, so how atoms are able to interact with other atoms to form molecules, like the biomolecules that we'll be talking about throughout this course. So again, with that, you don't need to learn anything for a test or anything. This is just for your information. But I hope that at the end of it, you'll have a really good understanding and be able to intuit how atoms will work based on just the periodic table of the elements. Then we'll practice looking at different biomolecules because we're going to be looking at these types of biomolecules for the entire course. So we should get familiar with them. And it's a lot of fun also to look at that. Then we'll talk about water and how important water is to life. So there's a few different interesting characteristics about water that make it really, really important for biochemistry. And then at the end, we'll talk a little bit about what we know as human beings about how life might have originated on Earth. So it's a really interesting lecture. I hope you all enjoy it. Again, I know it's a little bit long, but there's only one a week. And you don't have to remember anything for any tests or anything, just for your assignments and your short essay. So let's go ahead and get started. Lecture number 2, atoms, molecules, and life. So we're going to be talking today a lot about atoms and how atoms interact to form molecules, and basically the formation of all atoms can stem back to the Big Bang. So the Big Bang theory is the most widely supported theory for the formation of the universe. It's the most easily explained by the math and just observational evidence and research that's going on. And it basically posits that all matter originated at a single point, which is a singularity, that then expanded. It was basically infinitely dense over infinitely long time and then expanded, which formed subatomic particles, then atomic nuclei, then atoms, then protogalaxies and stars and heavier elements, etc. So here is a diagram of basically the timeline of the Big Bang theory from just, the less than a second after it occurred to present day. And so at this region right here, electrons were combined with protons and neutrons to form the first atoms, mostly just hydrogen and helium. So these would eventually coalesce based on gravity into stars and galaxies. And then eventually those stars made out of hydrogen and helium would condense and explode, and this released all the known atoms in the universe. So this brings us to the point we're at right now. So this is obviously an active area of research, and that is helped along with the new James Webb Telescope. One of the first images shown is right here. So this right here is a star. This right here is a star. This right here is a star. All the rest of those points are entire galaxies, which is really, really cool. So from this new James Webb Telescope, we're going to learn a lot about the universe in general. But the formation of the universe, the most widely accepted theory is the Big Bang theory, which resulted in the explosion of all matter in the universe and the formation of atoms to form protogalaxies and earliest stars, and then that eventually resulted in the formation of all the known atoms in the universe. So what is an atom? So an atom is the smallest unit of ordinary matter. Obviously, there's subatomic particles. If you break apart an atom, all sorts of stuff will fly out. But an atom is the smallest unit of matter that we basically think of matter. So what are atoms made out of? So atoms all contain a core nucleus, and that's formed from positively charged protons and uncharged neutrons, except for the case of hydrogen. And then negatively charged electrons are associated with that nucleus. So the number of protons will dictate the type of atom or element and help define its mass. So at the beginning of the universe, basically the only atoms around were hydrogen and helium. So hydrogen has one proton in its nucleus, and that is going to be surrounded by one electron for the most part. So that electron is going to be, it's usually depicted as sort of this, planetary thing where it's just revolving around the nucleus, but actually it's just present all around in a sort of cloud, and you can determine probabilistically using quantum physics where that electron is going to be, or what are the probabilities of that it's in a specific spot. So a hydrogen atom is made of a single proton and a single electron, so it has a mass of one atomic mass unit. So it's basically one just because it has that one proton. The nucleus is most of the mass of an atom. An electron is very small, and it's basically negligible when you think about its mass. Now, a helium atom has 2 protons, and it also has 2 neutrons in its nucleus. And so it has the mass of 4 atomic mass units because the protons and neutrons are basically the same size, or have the same amount of mass. So then that helium is the nucleus of the helium atom is going to be surrounded by 2 negatively charged electrons. So the positive charge and the negative charges have to cancel each other out, or you have a charged atom if there's more electrons than there are protons, or if there's less electrons than there are protons in the atom. So that's an atom. So it's the smallest unit of ordinary matter, and it's basically made up of these protons and neutrons in the nucleus, and those are surrounded by negatively charged electrons that stabilize or neutralize that charge. So stars are basically where hydrogen atoms and other atoms will coalesce via gravity to form these giant stars. So these hydrogen atoms will come together based on gravity, and they will release massive amounts of energy based on nuclear fusion. So the gravity is so strong in these stars that the hydrogen atoms will actually fuse. Their nuclei will fuse to form helium, which we just sort of saw in the last slide. So hydrogen atoms will fuse to form helium, and then more hydrogen can form with helium, and helium can fuse via nuclear fusion to form heavier elements. So all the heavier elements in the universe are formed in stars. So the first stars in the universe were called hydrogen giants because they were basically just made out of hydrogen. That was eventually fused into different elements, and then they can explode via supernova to release even heavier elements into the universe. So every single atom in the universe, every atom heavier than hydrogen or helium, was formed through this nuclear fusion process in stars, and the explosion of those stars via supernova. So stars, just like any other living thing, not that they're living things, have what's called a life cycle, basically. And for most of the life of a star, hydrogen is just fused into helium. So the gravity of the star will cause that hydrogen to fuse together, those hydrogen nuclei to fuse together to form helium. So the duration of a star, this is the most of its lifespan is just spent burning through that hydrogen into helium. Now, eventually, it'll sort of run out of hydrogen, and then the helium will fuse to form heavier elements, such as carbon. So helium, 3 helium atoms can fuse together to form carbon. So this is also an extended period of the life of a star. Now, after it runs out of helium, it'll start fusing these heavier elements, such as carbon, will be fused into oxygen, and this will occur for only a very short amount of period of the star. Then that oxygen will fuse to form silicon, and that's only 6 months of the life of the star. That silicon will fuse to form iron, and that only really occur for 1 day. So this is really the end of the life cycle of the star. And once it's formed into iron, that will cause what is a core collapse, basically. So this is when the star supernovas, and that only takes 1/4 of a second, and then that releases a massive amount of energy and material into the cosmos through the explosion of that star into a supernova. So iron is basically the end of the line when it comes to a star. It really has nothing else to fuse once iron, and then the core of the star will collapse, and it will release this massive amounts of iron and elements through the supernova, and massive amounts of energy. And that energy from the supernova will form heavier elements than iron. So really, you've heard it before, we're all made of stardust. We honestly are all made of stardust, because every element heavier than iron is formed from, and these lighter elements as well are formed in stars and then released through these massive supernovas. And so that brings us to the periodic table of elements. So the periodic table of elements really is one of the most important discoveries that humans have ever made. It's a visual representation of all the elements found in the universe, and it contains a lot of information regarding their atomic structure. So if you can understand what you're looking at when you're looking at the periodic table of elements, it really helps you understand chemistry in general and biochemistry in specifically. So elements are arranged on the periodic table in their order of mass. So here we have hydrogen, which has a mass of 1. It basically has 1 proton in its nucleus. Helium is right here, number 2. And then it comes down here. So it's arranged in these columns and these rows. And there's really, there is a rationale for them being arranged like this, which we'll get to in a bit. So we have lithium is element 3, beryllium is 4, boron is 5, carbon, nitrogen 7, oxygen 8, fluoride 9, neon 10, sodium 11, and so on and so forth for all of the elements known in the universe. So this is sort of a simplified version of the periodic table, because I'm cutting out the lanthanides and the actinides, which are usually listed below here. But we'll take some time and look at this periodic table of elements, and it will really help us understand how these atoms come together to form biomolecules important for biochemistry. So while the periodic table of elements lists over 100 different elements, all living organisms are mostly composed, and by mostly composed, I mean over 99% composed of just 10 elements, which are called the bulk elements. And so they're shown here on the periodic table in green. So we have hydrogen, which has a weight of 1. Then we have carbon. So that's the most important. All life is carbon-based, which we'll talk about here in a minute. We have nitrogen, which is 7, and we have oxygen, which is number 8. We also have sodium, which is number 11, phosphorus, which is number 15, sulfur, which is number 16, chlorine, which is number 17, potassium, which is number 19, and calcium, which is number 20. So all living organisms on the earth that we've ever come across, that we know anything about, are mostly composed of just these 10 bulk elements: hydrogen, carbon, nitrogen, oxygen, sodium, phosphorus, sulfur, chlorine, and calcium. So there are also 11 elements that are absolutely required for life for different living systems, but only in very, very small amounts. And these are called the trace elements, because they're absolutely required for living systems, but there's only tiny, tiny amounts in your body or the body of other living things. So there's 11 of these elements. So magnesium, which is number 12 on the periodic table, vanadium, which is 23, which is a metal. These right here are all metals. Manganese, which is number 25, Iron, which we mentioned is the heaviest element that can be created in a star directly. So number 26. Cobalt, number 27. Nickel, 28. Copper, number 29. And zinc, number 30. Also, selenium is required in trace amounts, so that's number 34. Molybdenum, which is 42. And then the heaviest element of the trace elements is iodine, which is absolutely necessary for life, but only necessary in very small amounts. So these 11 elements are required for life, but only in trace amounts. If you don't have one of these in your body, it can cause breakdown of living systems, but you only need very, very minuscule amounts. So less than 1% of your body is made of these type of trace elements. So we're talking about atoms, but really when we get into biochemistry and the rules of biochemistry, what's more interesting and what chemistry mostly looks at is the fact that elements interact with each other to form what are called chemical bonds. So via these two or more atoms will interact with each other by these chemical bonds to form what we call molecules. So these are specific chemical groups made up of two or more atoms that are linked together by chemical bonds. So the study of chemistry is basically examining the physical rules that govern which atoms interact to form which molecules and how these molecules can interact to modify or change or cause chemical reactions that change the atomic characteristics of these different molecules. So much of these rules on how these atoms and these molecules behave and how they form chemical bonds with each other can be deduced from the periodic table directly. So if you have an understanding of the periodic table and how it works, you can intuit how different atoms and molecules are going to interact with each other. So to get a better understanding of how these atoms interact to form molecules and how molecules interact to have chemical reactions occur, we're first going to ignore this whole region of the periodic table that includes what are called the transition metals. So these transition metals have their own sort of rules on how they interact, which is quite interesting, but not very important for biochemistry. So you saw that most of the transition metals are just present in trace amounts in your body. So these are a lot of times required to interact with other molecules or for enzymes to function. But the chemical rules regarding the transition metals, we can basically ignore in our study of biochemistry. Ignoring those transition metals, that allows us to basically have 1, 2, 3, 4, 5, 6, 7, 8 different rows left on the periodic table. Now, this is very, very important and it tells us how these atoms are going to interact to form molecules. So why this is important is because with the caveat of hydrogen and helium, all the other atoms are stable with 8 outer electrons. So we'll talk about more of this in a minute, but basically these atoms have electrons in the outer shell around the nucleus, and atoms are going to be stable when they have 8 electrons in that outer shell. So most atoms don't have 8 just as a single atom. They have to interact with other atoms to reach that rule of 8, which is actually called the octet rule, and to be stable. Now, the one caveat is hydrogen and helium, which we'll go into in a little bit. Helium is stable and it only has 2 atoms in its outer electron shell. So because these are smaller, they only need 2 to be stable. So hydrogen only has 1, but it can be stable with just 2 electrons in its outer shell. All the other ones, the larger electron shells, are going to be most stable with 8 electrons in their outer shell, which is the octet rule. So don't get too confused right away. Just understand the magic number 8. Basically, these are broken up into 8 rows, and then we'll go through how this works. And again, you don't need to memorize this or be able to use this, but it's interesting information to know. And it's really important if you want to understand or intuit how atoms and molecules interact with each other. So we can see already that there is this row right here, which is the eighth row that already fills in this octet rule. And so these atoms right here, helium, neon, argon, krypton, xenon, and radon, and we'll just ignore this sort of larger one because it probably has a different name than what's listed here. These are called the noble gases. And they're called the noble gases because they're already stable. They already fulfill that octet rule just by existing as single atoms. So they're noble gases because they don't really interact with any other atoms. They're very stable already the way they are. So we mentioned helium. So helium is number 2 here. And here is a depiction of a helium atom when we're trying to think about this octet rule. So helium is special because it only needs 2 atoms in this first valence shell to be stable. And helium, of course, has 2 protons, and therefore it has 2 electrons. So it's already stable. So this can be depicted like in this way right here with this helium atom. So this would be the nucleus, and then it has the 2 electrons surrounding that nucleus. So it's already stable just as an individual atom. Another way to write it is this way. So you just write He for helium and then put those 2 electrons so that you know it's stable. Now, if you move across this other row, the next noble gas that you get to is neon. So neon has 10 protons in its nucleus and therefore has 10 negatively charged electrons to stabilize that atom. So this is a depiction of neon right here. So we have 1, 2, and so those 2 electrons will fill that first valence shell. But there's going to be a larger shell around that shell to fit the other electrons that are going to stabilize those protons in the nucleus. So neon now has 1, 2, basically 3, 4, 5, 6, 7, 8, 9, 10. So we have those 2, and then we have 1, 2, 3, 4, 5, 6, 7, 8. So neon fills that octet rule in its second valence shell, and therefore it's very stable. So you can see neon has the number 10 right here, and it has 10 electrons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And so it can also just be depicted in this way where you're just showing the outer valence electrons. And you can see it has 8 there, so it's very stable just by itself. Neon doesn't need to interact with any other atoms because it's one of these noble gases. So we'll look at one more example of the noble gases. So that's argon. So it's right here. It's number 18. So it has those 10 electrons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then, but its number is 18, so it has 18 more electrons in the next valence shell. So 1, 2, 3, 4, 5, 6, 7, 8. So argon is also a stable noble gas. So you can just depict it like this, just showing those electrons in the outermost valence shell. So the noble gases already fill this octet rule without having to interact with any other atoms. But these are the only atoms, these noble gases, that do this, that fill this octet rule, because they're the only atoms that are already in this 8th row on the periodic table. So to look at how other atoms are not stable like the noble gases, we'll first need to interact with other atoms in order to fill that octet rule and become stable. We'll first look at hydrogen. So hydrogen, depicted here and here, only has 1 electron. So it has 1 proton and 1 electron. However, to fill that octet rule, or in this case, the rule where to fill that outer valence shell, it has to have 2 electrons to basically be stable like helium. So to do that, two hydrogens can interact, which you can show right here. And so what they'll do is these two protons will share these two electrons right here. So now both of these hydrogen atoms are now stable. So they fill that rule and they're able to be stable. So these two electrons can actually sort of exist all around these two protons in this hydrogen gas, which is just written as H2 because there's two hydrogen atoms in it. So this can also be depicted like this, showing that the hydrogens are sharing these two electrons. And what this is what's called a covalent bond. So it's a bond between these two hydrogen atoms because they're sharing these electrons in their valence shell, their outer valence shell. So that's why it's called covalent. So covalent bond. And so that is normally depicted when we show chemical structures, which we will over and over again in this class, as just a hydrogen with a covalent bond to another hydrogen, a single covalent bond. And so if you see this structure right here and you have some understanding of chemistry, you understand that it's two hydrogen protons sharing two electrons via this bond, and that's how they're able to be stable and fill their outer valence shells. So this hydrogen gas is very light. It's lighter than oxygen because there's still only two protons in it, and this was that's why it was first put into blimps, which actually ended in disaster with the Hindenburg disaster, because this hydrogen gas is actually very flammable, and that's why this blimp basically exploded for the most part. And so that's why we put helium into blimps now, because helium is stable just as itself. So those two, that bond can't be broken if there's external energy, because helium really isn't interacting with anything because it fills its outer valence shell. So that brings us to carbon, and carbon is the most important element when you're talking about life. So organic chemistry, the entire field of organic chemistry, is called organic chemistry because it's entirely based on carbon. So all known life is carbon-based. So you've probably heard that before, that all life is carbon-based. So why is that? So that's because of carbon's position on the periodic table. So carbon is in the fourth row and it has 6 electrons surrounding its nucleus. So we can see that depicted here. So here's a carbon atom. It has 1, 2, 3, 4, 5, 6. So it has 6 total electrons, and that means that it's missing 4 electrons to fill that octet rule. So carbon has to go 1, 2, 3, 4. It has to add 4 electrons to its outer valence shell in order to fill that octet rule. So carbon can also be depicted like this. So just the carbon, C for carbon, and then it has 1, 2, 3, 4 electrons in its outer valence shell. So carbon, you're not going to find carbon atoms just by themselves because they don't fulfill this octet rule. They need 4 other electrons associated with that carbon atom in its outer valence shell in order to be stable. So there's a very easy solution in nature to fixing this problem and making carbon stable. So we have hydrogen right here. All you have to do is basically make this element right here. So you can fill that outer valence shell of carbon by attaching 4 hydrogens to it. And this also, at the same time, fulfills the stability of the hydrogen, because hydrogen now is able to, and now it has 2 electrons in its outer valence. So now it's stable. So you can also show this molecule like this right here. So carbon, and then it's sharing these 2 electrons with this hydrogen. It's sharing 2 electrons with this hydrogen, and this hydrogen. So you can see this carbon now has 8 electrons in its outer valence shell and is stable. So the normal way to depict this would be something like this, where you just have a carbon and then you have single covalent bonds to 4 different hydrogen groups. So this is CH4. And so this molecule should be very familiar to you. It's methane. So this is it's a big component of natural gas, and it can be burned to provide energy in terms of methane. So carbon, all life is carbon-based because carbon is able to bond to 4 different groups in order to make itself stable. So we'll talk a lot about, basically entirely when we're talking about biochemistry, we're talking about carbon atoms because carbon atoms can bind to 4 different groups. So now we know that carbon can bind to 4 different groups to be stable, basically. However, that doesn't always have to be hydrogen. So that's a very common one, and that would be called a hydrocarbon. But carbon can also bind to other carbon atoms, as you can see in this molecule right here. So instead of just bound to 4 hydrogen atoms, carbon can bind to 1, 2, 3 hydrogen atoms, and then to another carbon atom that's bound to 1, 2, 3 hydrogen atoms. And you can see that fulfills the octet rule and the stability of all the atoms involved in this molecule. So you have a carbon right here, and it's bound to 3 different hydrogens. So that's fulfilling the octet and another carbon. And so that's fulfilling the octet rule of this carbon and the hydrogens. And then you have another carbon that also has fulfilled its octet rule. You can see there's 8 electrons surrounding these carbon atoms. So this would be commonly depicted as this molecule right here, which is called ethane, which can also be a component of natural gas. And it's C2H, it's H6, C2H6. So that would be ethane because there's 2 carbon atoms and 6 hydrogen atoms. So when you get really good and understand chemistry, someone can just show you C2H6, and then you know that that's 2 carbon atoms bound to each other, and then the rest of those groups, because we know carbon has to be bound to form groups, are going to be filled by those hydrogen atoms. So that molecule is ethane right there. So I'll go ahead and mention it right now, but you could also just draw a line, and an organic chemist would know that this is a carbon atom and this is a carbon atom. And because you know that carbon has to be bound to 4 things, you would just know that, oh, well, the other groups must be, the other groups that it's bound to must be hydrogen atoms. So these would be called hydrocarbons because they have a hydrogen and a carbon. And so these are going to be linked by this single bond here. So to make things a little more complicated, but just bear with me, carbons and other atoms can also form double bonds where they share more than just 2 electrons. So in a double bond, the 2 atoms are actually going to share 4 electrons in their outer valence shell. So 2 hydrocarbons can easily be linked by a double bond, as you can see right here. So in this case, you have a carbon, and it's actually double bonded. So they're sharing 4 electrons with this other carbon right here. And then these are going to be, if these are each bound to 2 hydrogens, it still fulfills the octet rule because you have 1, 2, 3, 4, 5, 6, 7, 8 electrons still surrounding that carbon atom. So this is a perfectly stable molecule, and it's called ethene. So we had ethane, that means it's a single bond. Now we have ethene, which already tells an organic chemist that there's going to be a double bond there just by naming conventions. So it can also be depicted like this. So you can see this carbon fulfills the octet rule because it has 1, 2, 3, 4, 5, 6, 7, 8 electrons around that carbon, and that's the same thing with this carbon right here. And so this would be depicted by an organic chemist just as this molecule right here. So there would be a carbon double bonded to another carbon, and then each of these are going to be linked to 2 hydrogens. So this would actually be C2H4. So this can also be depicted just by a line like this, and you would sort of intuit that there's a carbon here, and that each of those must be bound to 2 hydrogens to fill that octet rule. So again, you don't have to memorize all this stuff. Just understand that that by knowing the periodic table and knowing that carbon has to be bound to 4 groups, you can have things like single bonds between carbons or double bonds between carbons and single bonds with hydrogens. So it can get even more crazy, and you can actually have triple bonds in carbon as well. So carbon and other atoms can also form triple bonds. So in this case, there would be 6 electrons shared between the atoms in their outer valence shell. So we have this depiction of the molecule right here. And so right here, you can see there's 6 electrons that are being shared by these 2 carbon atoms. And then the other, number 7 and 8, would be shared with this single hydrogen molecule right here. So this could be depicted in this way, and you can see how it's fulfilling the octet rule. Even so, for this carbon right here, we have 1, 2, 3, 4, 5, 6, 7, 8 electrons around that carbon. So it would be more often depicted like this. So a carbon triple bonded to another carbon, and then to fill the octet rule, they each need to be bound to another hydrogen. So this molecule would be C2H2, and it would be called ethine. So we had ethane when it's a single bond, ethene when it's a double bond, and now we have ethine when it's a triple bond. So carbon actually there really isn't a huge limit to the amount of carbon, to the size of carbon chains that you can really make, because carbon can always be bound to another carbon via a single or double or triple bond, and that carbon can also be bound to another carbon. That carbon can also be bound to another carbon, that carbon, and so on and so forth, as long as it fulfills the octet rule. And so these hydrogens can actually be replaced with carbons, with hydrocarbons as well, and you can get branched chains as well. So these hydrocarbons can make really long chains, and this is actually very common. So we talked about lipids in the last lecture. So lipids are made, a big part of them are these long hydrocarbon chains. So up to 20 or more carbons is actually quite common in nature. So if you think of like a fat molecule, that's basically a carbon with an oxygen groups that are polar, but then there's a carbon, and you just have these big long chains that are made of these hydrocarbons. So each of these carbons are going to be linked to 2 hydrogens and then have single bonds with another carbon. So this can also be depicted right here, and you can see that each of these carbons fulfills the octet rule, or it could be just drawn like this. Although the more common way is to leave the hydrogens out and just draw a chain. Here's a 6 chain, and each of these are just assumed to be carbon. So this 6 hydrocarbon chain right here would be called hexane. And so this would be like a component of gasoline. So this is also a very energy-rich hydrocarbon bond. But I hope that I really just want to put across the lesson that carbon is a very versatile atom because it's able to bind to 4 different groups. So this allows it to do things like make these very long chains or these branched chains in biochemistry. So these hydrocarbon chains, the ones that we're most familiar with in our everyday life, are in the form of fossil fuels. So fossil fuels are basically natural gas, which is shown right here, bubbling up from the surface, oil, and coal. So all these materials are either gases or liquids or solids that are collections of hydrocarbons that were formed millions of years ago from the bodies of phytoplankton, zooplankton, and plants. So we talked about how all life is carbon-based. So most of the molecules in your body are basically carbon-based. So if you die and all the living things on the earth die and are sort of deposited in a specific area geologically, and then that's covered up with additional sediment over millions of years, that carbon in those living organisms, those hydrocarbon chains and those carbon atoms will turn into natural gas, oil, and coal. And so that's why when people talk about reliance on fossil fuels and switch to renewable fuels is so important, because these fossil fuels are not renewable. They were formed over millions and millions of years. They're very useful. They're transportable. They're energy rich. But once you burn them, they're not coming back until all life on Earth dies again and basically gets buried in the sediment to form new fossil fuels. But that's going to take millions and millions of years. So I hope this reiterates to you or emphasizes how important carbon is to life and how important it is these old fossil fuels are to our everyday life. And they're made up of these hydrocarbon chains that if you understand the periodic table, you completely will understand how they formed and why these atoms are able to interact with each other. So now that we've gone through carbon, we can discuss another element that is essential for life, nitrogen. So nitrogen is really ubiquitous in a lot of different biomolecules. So we have carbon right here. You can see that nitrogen is directly next to carbon. So nitrogen has one more electron, one more proton, and one more electron in its outer valence. So it's in the 5th row, and its atomic number is 7. So you can see nitrogen right here, and it has 1, 2, 3, 4, 5, 6, 7 electrons. And so that means nitrogen needs to fill 3 more electrons to fulfill the octet rule. So you can look at this on the periodic table as well. So 5, 6, 7, 8. So nitrogen, unlike carbon, which needs to bind to 3 other electrons, basically in its outer valence shell to be stable, nitrogen needs to bind to 3 other elements. So a common way that nitrogen is basically encountered is through nitrogen gas. So what nitrogen can do is it can already form a triple bond where the 2 nitrogens will share 6 different electrons. So right here we have 1, 2, 3, 4, 5, 6 electrons that are going to be shared between these 2 nitrogen atoms. And then we have 7 and 8, which would just exist on the nitrogen. So this can be, so those don't need to be bound to anything like hydrogen. Those are just electrons that are present in nitrogen. So nitrogen gas could be depicted like this, which fulfills the octet rule. You have 1, 2, 3, 4, 5, 6, 7, 8 electrons. And then the nitrogens are bound by a triple bond. And you could just draw those extra electrons, which would just exist on nitrogen here. And it would be called N2. So this is a common component of oxygen in our atmosphere. So actually, if you go scuba diving, there's going to be nitrogen gas in that tank. And it's an incredibly stable gas, nitrogen. So it's very hard to break that triple bond because it's so strong. So that's nitrogen. It's formed nitrogen gas. It's another important element to life. But instead of binding to another nitrogen through a triple bond, nitrogen can also bind to 3 hydrogens to be stable. So remember, nitrogen has to bind to 3 other elements, unlike carbon, which needs to bind to 4 elements. So nitrogen can just fulfill that by binding to 3 hydrogens. So that would be depicted right here. And you can see it fills the octet rule. We have 2, 4, 6, 8 electrons in the outer valence shell of nitrogen, and all those hydrogens are stable as well. So that could be depicted in this way as well. So nitrogen already has that, what it's called a lone pair. So these are a lone pair of electrons. So these are electrons that aren't bound to anything, but are just part of the nitrogen atom because it's got this extra 7th electron. And it can also be depicted in this way. And you could also put that lone pair right here if you wanted to. But the chemical composition of nitrogen bound to 3 hydrogens is called ammonia. So you've probably heard that term before. So ammonia is a key constituent of fertilizer, and it's actually what is incorporated into living things for nitrogen-containing compounds that are essential for life. So that difference between nitrogen gas and ammonia is actually really important because, as I said, nitrogen gas is incredibly stable. It has that triple bond, and it's really hard to pull that triple bond apart. So to actually incorporate nitrogen into organic compounds, it has to be converted from nitrogen gas to ammonia. And that process you might have heard of before is called nitrogen fixation. So you're fixing nitrogen gas into a form that can actually be incorporated into living things. All living things have nitrogen in it, and it's gonna be incorporated through this fixed nitrogen. So 25% of all the ammonium that is eventually incorporated into living things is produced through a process called the Haber process. And this was invented many years ago, I think in the early 20th century or even a little earlier than that. And it's basically an industrial process where you can take nitrogen gas and then incredibly high pressures and along with hydrogen gas, and you can actually turn that into ammonia that then can be used for fertilizer. So that ammonia can be incorporated into plants, and then we eat the plants, or cows or chickens or something eats those plants, and then we eat them, and that's how that nitrogen gets into our body to be able to use this. So before the Haber process was invented to make these fertilizers, the only ways to really get nitrogen fixation was through lightning and UV. So about 15% of the nitrogen in the living, organisms on Earth right now comes from just lightning and UV. So that provides enough energy to convert that nitrogen gas into ammonia. And then most of it, 60%, is through microorganisms that contain an enzyme called a nitrogenase complex. And so these microorganisms are associated with the roots of different plants that are able to fix nitrogen and basically take N2 and through this enzymatic process, convert it into ammonia. So you'll notice I'm actually from Ohio, and most of the farm fields in Ohio that are primarily want to make corn, but you can't just keep making corn because it depletes the nitrogen from the soil. So what they'll do is they'll basically one season they'll grow corn, and then the next season it'll lay fallow or something, and then the third season or the second season you will plant soybeans. And soybeans and other legumes have lots of these rhizobial microorganisms associated with these roots that are able to take nitrogen and convert it into ammonia that can then be incorporated into the biomolecules in the plants. So nitrogen fixation is a really important process necessary to make ammonia that can be incorporated, that allow that nitrogen to be incorporated into living organisms. So nitrogen as a component of biomolecules is usually actually attached to carbons. So since nitrogen has 3 electrons to fill in its outer valence shell, it can also form bonds with carbon atoms. So here we have a nitrogen, it's bound to one hydrogen, another hydrogen, and then it's got a single covalent bond with a carbon. And so this, along with this lone pair here, will fulfill the octet rule for this nitrogen atom. And also, since the carbon is bound to 4 different things, it also fulfills the octet rule for carbon. So this gives you this molecule right here, which is a carbon bond, single bound to 3 hydrogens, and then single bound to a nitrogen that then is bound to 2 other hydrogen molecule. So this would be CnH5. And so this molecule is actually called methylamine. So it's not like really a component of, it would be a building block for other biomolecules that we'll see that have single bonds between nitrogen and carbons. But it was mentioned in the television show Breaking Bad because they used this molecule to synthesize methamphetamine in that show. So, but in general, covalent bonds between nitrogen and carbon are very common, and you'll see them again and again when we start looking at biomolecules. And so the next atom that's really important for life is, of course, oxygen. So oxygen is also essential to living beings. Now, oxygen is right here in the 6th row of the periodic table. So it has 8 electrons. So if we look at this oxygen molecule right here, you'll see that by itself, it has 2 electrons in its inner valence, and then its outer valence, it has 3, 4, 5, 6, 7, 8 electrons. And so what that means is that it has 2 of these lone pairs that are perfectly stable, and then oxygen is able to bind to 2 other molecules. So carbon is able to bind to 4 molecules, nitrogen is able to bind to 3 different molecules, oxygen is able to bind to 2 other molecules. And you can see this again from the periodic table, so you just need 2 steps to fill that octet rule. Now, oxygen binding to other oxygen will form oxygen gas, which is shown here, but obviously these 2 oxygen molecules need to share 4 electrons to fulfill that octet rule. So it has 1, 2, 3, 4, 5, 6, 7, 8. So this fulfills the octet rule, but you have to have a double bond between this oxygen. So this would be shown right here. It's just oxygen, so it's just O2, and the form is a gas. And then you could even draw those lone pairs in these positions on the oxygen. So this is oxygen gas, which we all breathe and is exuded by plants and algae, of course, as a byproduct of photosynthesis. So plants and algae will take in CO2, use the carbon to form sugars, and then release that oxygen gas. We then burn carbon sugars, and the electrons from those carbons are actually donated to oxygen gas. So we'll talk about that in a bit more detail later. Now, of course, just like carbon and nitrogen, oxygen can fulfill that octet rule by just binding to single hydrogen molecules. So we have an oxygen here. It needs to fill these 2 positions on its outer valence shell, also shown right here. It can do that by binding to 2 hydrogen molecules, which gives you this molecule here that you should all be very familiar with, which is H2O, or water. So oxygen bound to 2 hydrogen atoms forms H2O, which is water, and can be depicted in this way here. So it's oxygen with a single bond to 2 other hydrogen atoms, and it would have these 2 lone pair electrons that are already associated with oxygen at these positions right here. So water is, of course, essential to all living things, and we'll discuss that later in the lecture specifically. And of course, just like nitrogen, oxygen can also form single bonds or double bonds with carbon. So we have this molecule right here. So oxygen can have a single bond to a hydrogen molecule, and then the other single bond can be to another carbon molecule, or in this case, a hydrocarbon. So then the oxygen here has its octet rule complete, bound to 2 different species, and then carbon is bound to 4 different species. So carbon is also stable in this form. You can see here, both the oxygen and the carbon and the hydrogens are all fulfilling their octet rule and are highly stable. So this molecule would be depicted right here with single bonds between the carbon, the oxygen, and the hydrogens. It would be called CH5OH, or actually you call it CH3OH. I don't know why that's there. So this group right here would be called a hydroxyl group. And you can see why it's named that hydroxyl, hydrogen and oxygen group. So a hydroxyl group, or an alcohol group is the other way you would call it, is bound, when it's bound to a single carbon, that gives you the alcohol methanol. So this would be called methanol right here, just like methane, but since it's an alcohol, you call it methanol. So if there were two carbon groups here, so we have CH3, another CH2, and then an OH, this molecule right here would be the familiar molecule you may be familiar with, called ethanol, which is the alcohol that you think of in liquor and beer and wine. So oxygen bound to carbon is also a very important molecule, important to life's processes. And carbon can also really often forms double bonds with carbon as well. So you can see this molecule right here. It's a carbon, and then it has a double bond, so it's sharing 4 electrons with this oxygen here, and then it would, to fill its octet rule, would need to be bound to 2 hydrogen molecules here. So this could also be shown right here. You can see the oxygen and the carbon are both fulfilling the octet rule. And but this would more commonly be depicted in this way right here, where the carbon is a double bonded to this oxygen and then has these single bonds to these hydrogen groups. So this would be CH2O. And so this would commonly be depicted in this way right here. And this molecule is actually called formaldehyde. So these are formaldehyde groups. So this carbon can actually be attached to other carbons and stuff like that. But formaldehyde, which you probably heard of as a common preservative and fixation material. So oxygens can form double bonds as well, and these are very common. You'll see them again and again in the organic compounds that we look at in this class. Now, another common atom that's a little bit less common than carbon, nitrogen, and oxygen. So if you just have carbon, nitrogen, and oxygen, you can make all sorts of different biomolecules that are just made out of those 3 elements. But sulfur is also found in a lot of different biomolecules, especially in amino acids that make up proteins, which we'll look at in the next lecture. So you can see that sulfur is in the same exact group, same exact row. So it's in the 6th row, just like oxygen. It's just 1 row below. And so that means it has basically 1 valence shell, 2 valence shells, and then it has a 3rd valence shell. But that 3rd valence shell is going to look a lot like oxygen, where it has these 2 lone pairs, and then it needs 2 more electrons to fill the octet rule. So it's going to behave very similarly compared to oxygen. So it can also form these single bonds with carbon. As you can see right here, it just has 1 more valence shell in its outer valence shell. But you can basically depict it the exact same way as you would oxygen, and with these single covalent bonds right here. So this would be CH3SH. And so, so sulfur behaves very similarly to oxygen. It just needs to bind to 2 different atoms to form organic compounds. And these are very common in amino acids. The one difference is that carbon really doesn't often form double bonds with sulfur, and that just has to do with the fact that sulfur is a lot bigger than carbon. And so those types of double bonds are a bit less stable, just because the outer valence shell is, a larger valence shell. But that's not a detail that you need to worry about. Sulfur can basically be intuited similarly to oxygen because they're on the same row in the periodic table. And now if we also look at this molecule right here, so chlorine, so this isn't exactly a common atom incorporated into biomolecules. So that's why I wrote up here covalent bonds of life with an asterisk, but it is important in a lot of different organic compounds like drugs and stuff like that. So chlorine can also form single bonds with carbon, but you can see that chlorine, its outer valence shell is full except for only 1 position. So chlorine is really only going to form a single bond with something like carbon to fulfill its octet rule. So it's only missing 1 electron. So if you just form a single bond, a covalent bond between a hydrocarbon and chlorine, that will fulfill the octet rule of all the atoms involved. And so that would be shown right here. So this isn't a really a molecule that you would find in living things. So we'll get to what chlorine or chloride does in living systems in a minute. But just to demonstrate the fulfillment of this octet rule with these other atoms that are bound to carbon. So the last molecule that we're going to talk about over in this section here is phosphorus. But phosphorus is really important to biological systems, but it's mostly just found in the form of phosphate. So it gets a little more complicated when you think about phosphorus, because it's only found in this form where phosphorus is bound to 4 different oxygen groups. And so this is commonly depicted as this molecule right here. And it's basically phosphorus double bonded to an oxygen and then with single bonds to 3 other oxygen groups. It's not super important that you understand the details of this, but it does carry negative charges on these different oxygens. So, and that's because of these different groups here. So actually, I believe it's 2 negative is the group on phosphate. So this double bond, these electrons, while it's depicted as this double bond, single bond, it's actually these electrons are sort of shared throughout the whole phosphate molecule, giving it a 2 negative charge. So don't get too caught up with phosphate or phosphorus or anything like that, but do remember this phosphate molecule because it's so important for biomolecules. So phosphate, because it carries these negative bonds, it's very important for a lot of different biological processes. So we've already mentioned a little bit about this molecule ATP. So this is actually a nucleic acid, and it's the universal energy currency used for all living systems. And what I mean by that is just a very energy-rich molecule that you can use to drive chemical reactions that require an input of energy. So if that didn't make any sense to you, don't worry too much about it. So this is ATP, and basically it has these phosphate groups here. And it's an energy-rich molecule because these are very unstable. You can basically break one of these phosphate groups off and release it, and that releases energy. So these phosphate groups are really important to transform energy in your body. Another way that phosphate is really important is because it's a huge component of DNA. So DNA is made of nucleic acids, just like this ATP molecule here. And the phosphate group is actually that all these outside groups here on the DNA double helix are phosphate groups. So they actually form bonds between adjacent nucleic acids to form these polymers, and it has a negative charge on the outside of this DNA molecule. So phosphate's important for that. And the last thing that phosphate is really important for is that it can be added onto proteins to sort of change the structure or the function of that protein. So here we have a protein, and then you have this amino acid sticking out. Well, it's got this OH group, and you can stick a phosphate molecule on the end of that. So we'll get into this a little bit more later, but that can basically make it so the protein is turned off when there's no phosphate on it, and then it gets phosphorylated, and now that protein actually has activity. Or the protein can have activity, and you take the phosphate group off, and now it doesn't have any activity. Stuff like that. So phosphate is really important. Phosphorus really only occurs in this form of phosphate when we're talking about biomolecules. Now, one more little caveat when we're talking about covalent bonds between molecules. So we've been depicting them as sort of planar groups. So we talked about methane before, and we sort of just drew it like this. So carbon bound to 4 hydrogens. But if you think about it, if you think about things energetically, basically this planar way is just a way to write it on a piece of paper. So in reality, it would form a tetrahedral form where these hydrogens are going to be sticking out in a pyramidal shape because those hydrogens need to be as far away from each other as possible. So you just think about that a little bit, but that's basically how molecules are arranged. And sometimes when we draw molecules, it sort of takes this tetrahedral orientation into account. So we have one hydrogen that's sticking up, one hydrogen that's sticking towards us off the page, one hydrogen that's sticking away and to the right, and then one hydrogen that's sticking towards us, sort of to the side. So this would be a tetrahedral sort of orientation. And this does become quite important. So if we think of ammonia, which is shown right here, so it has this lone pair of electrons, and then it's bound to 4 hydrogens. So really that lone pair is going to be sticking this direction, and then these hydrogens are going to be sticking as far away from each other as possible. And that's just the most energetically stable way for this molecule to exist. The same thing with water. So water has this sort of bent orientation, and that's because it has these 2 lone pairs. So those electrons are going to be sticking out far away from each other, and then the hydrogens are going to be as far away as possible from those lone pairs. The same thing with phosphate that we just talked about. So phosphorus is going to be bound to 4 different oxygens, and just because it's the most stable, those oxygens are going to be pointed in this specific tetrahedral orientation as far away from each other as possible. And that's because they each have electrons that are going to sort of repel each other on each of these oxygen atoms. So it's not super important, but if you've ever played around with like the ball and stick models, and really we should probably do that in the lab associated with this class, but it makes perfect sense that these atoms are going to be as far away from each other as possible on these bonds. But for all intents and purposes, usually we just write it on the paper in this sort of flat orientation. You just need to understand, the more you learn about chemistry, it's just assumed that those atoms are going to be pointed in the most stable way. They're going to exist as far away from each other as physically possible. Now, there's one more way that carbons interact with each other, or hydrocarbons interact with each other to form molecules that we haven't really touched on, but I have shown you some examples of. And this is the form of benzene, which are also called aromatic or aromatic compounds with phenyl groups as well. So benzene is shown right here. And this is sort of a special case, it's a special way that carbons can interact with each other in a ring. So I already showed you the sort of chain that carbons can make, or hydrocarbons can make. This is a way for carbons to interact that's really stable and forms a ring structure. So what is happening here is that there's 6 different carbon molecules interacting with each other in a ring here. But each of those 6 carbons is actually only bound to 1 hydrogen. So how is this possible? A carbon is supposed to be bound to 4 different things, but if you look at this carbon, it's only bound to 3 different things. Well, how this works is that there's double bonds associated with these different carbons. So you can see here there's 3 double bonds depicted in this structure. So actually, like this carbon here, because it's got this double bond, is bound to actually 4 things. So it's carbon 1 to the hydrogen 1, and then double bound to the carbon, so that's 4. So that fulfills the octet rule for that carbon. But in reality, this double bond isn't right here. This double bond is actually moving all the time. So that double bond can go there, and then it can move here, and then it can move here, and then it can move here, and then it can move here. Those extra electrons are actually being shared throughout the entire molecule. So this type of molecule, so benzene, was first figured out by this guy named August Kekule, who I believe was a German guy. And what his story, at least, that he told was he had a dream, because people were confused how you can have this 6-carbon ring with only 6 hydrogens. You should have 12 hydrogens associated. Well, he had a dream where he dreamed about what's called an Ouroboros, which is a snake that's basically eating its own tail. And that's sort of how this benzene ring works. So they're often depicted in this way right here. So you have the 6, each of these are carbons right here. You have 6 carbons, and then there's a double bond, 3 double bonds that are shared between this entire molecule. So this would be what could move to this bond, this could move to this bond, this could move to this bond, and then you'd have that right here. So it's often actually depicted as a 6-membered ring with just a circle in the middle, meaning that these electrons, those extra electrons that are necessary to fill the octet rule, are shared between those 6 carbons. Now, this type of group, so this is actually called a phenyl group when it's attached to something else. Like in this molecule here, you can say, oh, there's a phenyl ring, or you could say a benzene ring, or you could call it an aromatic ring. And the reason why you call it an aromatic ring is because these types of rings are really common in molecules that have a specific scent to them. So this molecule here, cinnamaldehyde, that is what basically makes cinnamon smell and taste like cinnamon. So you can see it has this benzyl ring right here, or this phenyl ring right there. I'll use those terms interchangeably. Vanillin is another one. You can see it has this ring here. And that's what gives vanilla the smell and taste that it has. Thymol has this ring right here. That's what gives the herb thyme its smell. And then eugenol is another one. So that's another molecule that has one of these benzene or aromatic rings right here. Eugenol gives the scent and flavor to a lot of different things, including cloves. So these benzene rings, just give that a little bit of thought. It's basically 6 carbons, you can see here. And then it's depicted with these double bonds, but really these electrons are being shared by all 6 of these atoms. And so these are named also aromatic rings because of just the history of chemicals, and some of the first chemicals that were investigated and studied were like what makes cinnamon smell like cinnamon, and so on and so forth. So we'll come across these a lot when we're looking at different biomolecules and structures of organic compounds. So now that we've talked a lot about the rules that govern how different atoms are interacting to form molecules, we can start looking at chemical structures, because we're going to show a lot of chemical structures in this class. You don't need to understand them in any detail. You don't need to memorize any of them, but it can be quite fun to look at the different chemical structures. And if you can understand a little bit about how the differences and the similarities between different chemicals, that'll help you, just understand. It'll help you get way more out of this course than you would otherwise. And when you ever see a chemical structure, it'll make a lot more sense to you. So we'll just practice looking at a few different chemical structures and just see what information we can glean from them. So these structures below are all what's called amino acids. And so we'll talk about these a lot more in the next lecture, but these are individual chemicals that can combine and form polymers that make up proteins, which are really important, obviously, biomolecules. It's a whole class of biomolecules. So each of these structures below have a certain feature in common, and that feature is shown right here. So this is the amino acid serine, this is the amino acid cysteine, this is the amino acid leucine, and this is the amino acid phenylalanine. And so this region that I just circled is exactly the same for each of these different amino acids, and it actually gives them their name. So the amino from amino acid groups comes from this group right here. And so this is a nitrogen bound to 2 hydrogens and then bound to a carbon that's bound to other stuff. And so that group here, this group here, is called an amino group. And that comes from the word ammonia, which is, of course, that NH3, where the nitrogen is bound to 3 different hydrogens. So each one of these amino acids has this exact same amino group right here. So the other part, the acid part, comes from this part right here. And so this part right here is called a carboxylic acid. So let me write that down. Carboxylic acid. And so this is a carbon right here. So when we're looking at these structures, we don't always write, usually you don't write the C's for carbon. You just have a line there, and then it's assumed that it's a carbon. And that's because carbon is so common that you don't need to write the C every single time. So this carboxylic acid group is a carbon double bonded to one oxygen, and then with a single bond to another oxygen that's bound to a hydrogen. So we'll talk about more of this later in the lecture, but that hydrogen can actually be removed from there, and that proton actually makes solutions acidic. So the acidity is a measure of how many protons are in a solution. So you can see that all of these structures have this carboxylic acid group right here. And actually, this double bond is a lot like benzene, where it can actually be shared between these two groups, but that's not super important. Now, what's different about each of these amino acids is the group sticking out on this side here. So what groups are sticking out on each of these sides? Well, in serine, we have a carbon group right here, and you can see that the C is not written, and also the hydrogens aren't written. There's actually two hydrogens sticking off of this carbon, but we don't write them in the structure because you just know that carbon is bound to 4 different things. So it's bound to 1, 2, 3, 4. And then in the serine, it's also bound to this OH group, which we remember is an alcohol group or a hydroxyl group. So that's serine. Cysteine is very similar to serine in that it has a carbon, but then it's bound to a sulfhydryl group is what you would call it. And you remember that oxygen and sulfur both can be bound to 2 different things. And in both these cases, they are bound to do different things. So one is a carbon and one is a hydrogen there. And this one's bound to a carbon and a hydrogen here. So serine and cysteine are very similar, just in the same way that oxygen and sulfur are very similar in the way they bind to things. So this molecule here, leucine, you see that it has a carbon, and then that carbon is branched, and it basically just has 4 carbons. And again, we don't write the hydrogens on here because you just assume that there's 3 hydrogens there because carbon has to be bound to 4 things, 3 carbons there because carbon has to be bound to 4 things, 1 hydrogen here because it's bound to 1, 2, 3, 4, and then 2 hydrogens here because it's bound to, we know that that carbon is bound to 4 different things, so we don't write that. And then the last amino acid I have listed here is phenylalanine. And so the phenyl in that name comes from this phenyl or benzyl ring right here. So it has a carbon right here, and that would be bound to 2 hydrogens. And then it has this phenyl or benzyl ring here. And we just know, because we understand how benzene works, that it's going to be bound to 4 hydrogens. So sorry that got kind of messy, but we're just practicing and having some fun looking at chemical structures. So here's some more chemical structures of different what are called analgesics or pain medicines. So there's lots of different groups that we can look at on these different molecules. So we have aspirin right here. So it has this benzyl ring right here, and it's attached to this group right here, which we already determined was a carboxylic acid group. So it's a carbon double bound to an oxygen and then to a single OH group. The aspirin also has this group right here, which is called an ester group. So this phenyl ring right here, instead of just having 4 hydrogens, it's bound to this carboxylic acid group. And in this one, it's bound to an oxygen, which is bound to another carbon that's double bound to an oxygen. And then here is another carbon at the end. So this is actually called an ester group, but that's not very important for you to remember or anything. You can see ibuprofen right here. It's a sort of a similar structure. So what does it have going on here? So it has a benzyl ring right here. And then it, just like leucine, it's got this hydrocarbon chain, and they just happen to write the CH3 group. So sometimes people write those, sometimes they don't. You see, they didn't put the C here, but they put it at the end. They didn't put the C here, but they did put it in these 2 positions here. It also, this phenyl ring is also bound to this carbon, which is bound to this carbon, and is bound to this carboxylic acid group right here. So that's ibuprofen. And then we have acetaminophen, which is the active ingredient in Tylenol. So that also has a phenyl ring. You can see how common these are. And it's bound to an alcohol group or a hydroxyl group. And then it's bound to a nitrogen. And nitrogen, of course, needs to be bound to 3 things. So it's bound to a hydrogen. And then it's bound to another carbon that's double bound to an oxygen. and then bound to another carbon. And again, you just assume that these 3 hydrogens are present on that one. And then lastly, we have the most powerful of these pain meds, morphine. You can see it has a very complicated structure. It's got 2 phenyl rings that are bound to an oxygen here, that's bound to another carbon, that's bound to another sort of ring, but it only has 1 double bond. And then these lines mean that this hydroxyl group is going away from us, and then this carbons are coming towards us, and then this nitrogen is right here. So this is a very complicated structure of morphine, which binds to opioid receptors in your body. So there's just more chemicals, low stakes. We're just practicing looking at them and seeing what information we can get out of them. And now another important point is you can understand how some drugs might work, whether, they're pain medicines or even recreational drugs that are used recreationally by some people, based on their similarity to the natural compounds that they sort of mimic. So right here, we have this molecule epinephrine, which is also called adrenaline. So this is the molecule that's secreted by your body in a sort of fight or flight response. So if you're walking down the street and you see a lion's looking at you and it's going to start chasing you, your body's going to flood your bloodstream with epinephrine or adrenaline. That's going to bind to cells and basically make it so you could run away really fast. Well, you can see epinephrine has this phenyl ring here. It's got these hydroxyl groups. It's bound to this carbon, and then a nitrogen group, and then another carbon here at the end. Now, if you take a drug like amphetamine is a component of some like ADHD medications. It's also used recreationally, called speed. Methamphetamine is sort of similar to it structurally. You can see that amphetamine has this same sort of phenyl ring and then a similar carbon, but there's no hydroxyl groups on it. Then there's another carbon, then there's a carbon sticking off right here, and then there's that nitrogen group. So it has a pretty similar shape. And so it's actually not very surprising that amphetamine can bind to some of those same receptors that adrenaline or epinephrine does. And that's why it will sort of pump you up and make you more awake and alert and stuff like that. So that's what it does to most people. And it's because it's similar to epinephrine as a molecule and can bind to the same receptors. The same thing with serotonin. So you can see serotonin has this interesting phenyl ring, and then another ring that contains a nitrogen group. It's got this OH coming off here, and then it's got carbon, and then an amino group right here. So the molecule that's in what are called, magic mushrooms or psychedelic mushrooms is psilocybin. And you can see it actually, although it's not really completely understood how it has the effects that it does, it can actually bind to the same serotonin receptors, which affect how your brain functions. You can see it has this same sort of structure right here. It also has this nitrogen group here, although it's got some carbons sticking off of it. And then it's got this phosphate group right here. So by understanding chemical structures, you can sort of understand how some drugs can mimic natural components of your body and have similar, although different, effects. So now that we've gone through covalent bonds, we can look at a different type of bond that's also really important to life and that we can also understand through our understanding of the periodic table, called ionic bonds, or sometimes called electrostatic bonds. So these occur between atoms that are actually charged or become charged after gaining or losing electrons. So instead of sharing electrons on these as a covalent bond through the outer valence shells, these atoms will actually interact by and fill that octet rule by gaining or losing electrons. So a really common example is sodium and chloride, which are both important for life's processes. So sodium, you can see, is here in the first column of the periodic table, and chlorine is in the 7th row of the periodic table. So that means what's going to happen is that, so sodium has only one extra electron in its outer valence shell, and chlorine only needs one electron in its outer valence shell. But instead of sharing those electrons in their outer valence, what's going to happen is that sodium is going to lose that electron and become positively charged. And then chlorine is going to gain that electron. So that electron from sodium is going to be donated to chlorine, and it's going to carry a negative charge. And because sodium is now positively charged and chloride is now negatively charged, that gives us a sodium chloride salt. So that's what a salt is basically. It's a positive or negative charged compound that interacts with another positive or negatively charged compound. So this gives us sodium chloride, which is table salt, and that could be written as sodium positive chloride negative. So this is very common. These are very common, commonly involved in biological systems, especially neurologic systems. Another really common one is potassium chloride salts. So not all salt are sodium chloride. They can also form from these other atoms. So potassium is just below sodium on that first column. And so potassium, while it has another valence shell, it also just has that one electron. So it can also interact with chloride very easily just by losing that electron, donating it to chloride. And now you have potassium with a positive charge interacting via this ionic bond with chloride that carries a negative charge. So this gives you the salt of sodium or a potassium chloride. And then the last atom or element that's really important for life is also calcium. So calcium, of course, is really important for bone and tooth formation and development, and it can form a salt with chloride as well. But you'll notice that calcium is actually on the second row of the periodic table. So what does that mean? It means it has 2 extra electrons in its outer shell. So it'll actually donate one of those electrons to one chloride and then another of those electrons to another chloride. So calcium chloride salts are always going to be CaCl2. So this would be the depiction right here. So calcium's going to carry actually a 2 positive charge, and then that is going to interact with these 2 chloride ions. They're called ions when they're positively or negatively charged. So cations are positively charged and anions are negatively charged. So calcium chloride would be another example of a salt, although it's a little bit different from sodium chloride and potassium chloride. So we also mentioned that there's these 11 elements that are also necessary for life in very, very small amounts. So we said magnesium, vanadium, manganese, iron, cobalt, nickel, copper, zinc, molybdenum, selenium, and iodine. So these are absolutely essential for different forms of life, but they're only necessary in very small amounts. So a very good example of one of these is iron, which its chemical name is Fe, or its chemical symbol. So this molecule is really important for your body. It gives your blood actually its red color. So iron will be bound in the middle of a molecule called a heme molecule, which is shown right here. So this whole molecule right here is called a heme molecule. And so it's this cyclic ring made of carbons and some nitrogens that coordinate this iron atom in the very middle of it. And this heme will actually be bound to a protein. So this protein right here is myoglobin. It's very similar to the molecule hemoglobin that you might have heard of as well. So what hemoglobin and myoglobin do is they will transport oxygen gas throughout your bloodstream by binding this oxygen to the iron molecule. So it'll bind that molecule and it'll release that molecule when it gets to cells in your outer tissues that require oxygen for basically respiration. So iron is a very good example of how important these trace elements are in life's processes. Another really important trace element is iodine. So it's actually the largest element that is required for living systems. So it is important because it's actually bound covalently to this molecule right here to form what's called thyroxine. And so this is a hormone that's really important to thyroid function. So your thyroid gland is an endocrine gland that's basically located in your neck. So the absence of iodine in your body basically causes your thyroid to become enlarged, and you can get what's called a goiter. So this individual right here has a goiter, which is basically the enlargement of your thyroid line in your neck. It can also cause all sorts of other developmental problems. So this is the reason that usually when you buy salt, it's iodized. It's a really easy way to get trace amounts of this iodine into people's diet so that they don't develop these goiters. So it first became available in the US, and it's subsequently available all over the world. But it first became available in the 1920s because in certain geographic regions like the Great Lakes and the Pacific Northwest, there's not a lot of iodine in the soil or in the foods that people eat, and people were getting goiters all the time and all these sorts of disorders. So that's why salt, you could basically make salt available that has iodine in it, and you can just get the trace amounts of elements of iodine in your diet just by, putting salt on your food. Now, most of these other trace elements are just used by specific enzymes to help them catalyze chemical reactions. So enzymes, of course, are proteins. So here's an enzyme right here. These are proteins that catalyze chemical reactions. So they can change one chemical into another chemical, or they can take two chemicals and combine them into one chemical, stuff like that. So a lot of different enzymes, the way that they catalyze those chemical reactions is by coordinating the protein with these sort of trace elements here. So an example of this would be the enzyme carbonic anhydrase, which is one of the fastest enzymes known. And it requires small amounts of zinc, which is one of these trace elements, which is number 30 on the periodic table. So it can't catalyze those chemical reactions without zinc being present in the active site of this enzyme. So you don't need much of it to function, but you do need small amounts of zinc. So there was the old tagline from this multivitamin, they might still use it, called Centrum, where they said it's a multivitamin from A to zinc. And that's because you need small amounts of zinc in your body to function properly, including this carbonic anhydrase enzyme. So now we can start talking about the molecule that is central to all of life and all of biochemistry, and that's water, which is H2O. So all biochemistry and all of life processes basically occur in an aqueous environment. So your body is mostly made out of water, and all the interactions between all the chemicals in your body that sustain your life are occurring in water, in an aqueous environment. So there's a lot of different reasons of this, but the main reason that it all boils down to is that water is a polar molecule. So we showed earlier how water is H2O. Oxygen has to be bound to two different things, and then it's got these two lone pairs of electrons. Well, what the result of that is that oxygen is really electronegative, or you can think of it as really greedy. So most of the electrons, including the ones that are shared with hydrogen, are going to be pulled towards this oxygen molecule and away from these hydrogen molecules. And what that means is that you have a partial negative charge on that oxygen and a partial positive charge on each of those hydrogens. So this delta signal, which is like a lowercase Greek d, means partial charge. So it's not a full positive or negative charge, it's a partial positive and negative charge. So this is depicted really well right here. So most of those negatively charged electrons are going to be surrounding this oxygen atom, and that's going to leave a deficiency in electrons around these hydrogen atoms. So you have this partial negative charge around the oxygen and a partial positive charge around the side of the hydrogen. So what that means is that this negative charge can interact with other positively charged stuff on this side, and this partial positive charge can interact with negative charges on this side. So this makes it polar. So it has two poles. So one side of the water molecule is going to be more electronegative, have more electrons that can interact with positively charged species. And the other side has a slight positive charge. It has less electrons around the hydrogens. So, this electronegativity of the oxygen in the water molecule and the electropositivity of the hydrogen molecules allow it to form what are called hydrogen bonds with other molecules, including other water molecules. So, you can see here is H2O. The oxygen is partially negatively charged and the hydrogens are partially positively charged. Well, this negative charge on this oxygen atom can therefore form weak bonds with this partial positive charge on the hydrogen molecule of another water atom. So these are two different water atoms. So this is a partial positive charge, this has a partial negative charge, and that allows them to interact with each other because positive charges are attracted to negative charges. And you can see in pure water, there's going to be all sorts of these interactions between these partially positively charged hydrogens and the partially negatively charged oxygens. And this is a really important component of how water functions. So this interaction between water molecules is what gives water surface tension and why this bug here is able to basically walk on top of water. It's why if you do a belly flop in a water, it hurts. It feels like you hit a concrete wall, and that's because of these strong interactions in liquid water. It's also what allows these huge, tall redwood trees to push water all the way 200, 300 feet up into the tops of the leaves from the soil. So this gives it surface tension, and it allows it because of these hydrogen bonding interactions, so these weak bonds between these different water molecules. So these interactions between these water molecules in liquid water also makes water highly concentrated. So we measure the number of atoms in a sample or a solution is measured in what's called moles. So 1 mole equals 6.02 times 10 to the 23 atoms. It's a ridiculously large amount of atoms in 1 mole. So that's 6 with 23 zeros after it. There's not even really a name. It's like a gajillion, basically. You just make up a name. But it's 6 times 10 to the 23 atoms of the pure molecule. So we measure the concentration in a solution. That is how many atoms are in a solution in terms of molarity. So that 1 molar is 1 mole in 1 liter of solution. So if we think about water, so 1 mole of water is 18 grams because hydrogens equal 1. So you got 2 hydrogens, so that equals 2. And then, that's another one. And then oxygen, the molecular weight of oxygen is 16 then. So that gives you 18 grams. So 1 liter of water weighs 1 kilogram or 1,000 grams. But overall, the concentration of water in 1 liter is 55.55 molar. So there's a lot of different water molecules in a sample of pure water. So that's a very important feature. And most biomolecules are not present in a solution at that high of a concentration. Another really weird part about water. So it's been said that if we didn't know about water and we discovered it on some distant planet or something, we'd be like, this is quite a strange. This molecule has some strange properties to it. So one of the things, and that's also important in life, is that water, unlike most materials, is actually less dense as a solid. And this just has to be due to the interactions between water molecules at lower temperatures. So in ice or solid water, of course, the hydrogen bonds are basically very stable and they form these crystalline forms and they line up really nicely. So the hydrogen's interacting with the water. But in liquid water, these are constantly breaking and forming. And so this actually causes water to be more dense than ice and allows ice to float on the surface of water. And this, of course, is very important to life. If you think of all marine life, they basically depend upon the fact that if water freezes, they can still exist below that ice that's on the top of water. So this is really essential to all marine life on planet Earth. And the fact that that ice is less dense than liquid water is also actually also has some other consequences because it expands upon freezing. And this can cause your cells, which are made mostly of water, to rupture when they're frozen. So this is responsible for both the death of plants after a frost. So if you've gone outside after a frost, that the water in those plants will actually expand and then cause the cells in that plant, those plants to burst open, basically. And so that's what causes the plants to die. It's also what causes frostbite that can basically kill your cells on your extremities if your body gets too cold. And then you're left basically, having to amputate toes and stuff like that because it's just dead tissue, basically. So, the fact that water is less dense as a solid is important in marine life, but also has consequences for other forms of life. Another really important feature of water is that it remains liquid over an extended range of temperatures. So those strong interactions, they're weak interactions, those hydrogen bonds between water, but because water is so concentrated, all those weak interactions add up, and it basically makes water very stable as a liquid. So water remains a liquid over an extended temperature range. So it freezes at 32 degrees Fahrenheit and then boils, turns into a gas at 212 degrees Fahrenheit. So this range happens to correspond to the general temperature of the planet Earth. So that's why when scientists are looking for other planets that may be habitable to life, they look in what's called the Goldilocks zone. And this would be a region far enough away from a star, but also close enough that water would be able to remain liquid. It'd basically be in this range from 32 degrees Fahrenheit to 212 degrees Fahrenheit, or 0 degrees Celsius to 100 degrees Celsius. So this is also another really important feature of water for sustaining life. Now, this ability for water the fact that water is polar is also really important for biochemistry in general when you think about how it dissolves other compounds. So a lot of biochemical compounds are also polar, and they can form hydrogen bonds with each other, and they can also form hydrogen bonds with water. So we have a water molecule here. Of course, the oxygen is electronegative, and the hydrogens are partially positive or electropositive. So if you have other molecules like this carbon double bonded to this oxygen, which we already saw was really common in these biomolecules, this oxygen is also going to be electronegative, and it can form hydrogen bonds with water because this hydrogen on it is electropositive. The same thing with like nitrogen on different compounds, which we already saw was a component of a lot of different compounds. If it's bound to a hydrogen, then this biomolecule is electropositive, and it can form a hydrogen bonds with electronegative oxygens on water. So another important thing that's a little less related to water is the fact that these two molecules can form hydrogen bonds with each other. That's really important for a lot of different biomolecules, including these nucleic acids that form your DNA in your body. So you have A's, T's, G's, and C's, and those interact in the DNA double helix through these hydrogen bonds between these different atoms. So this nitrogen hydrogen here is electropositive, this oxygen is electronegative. They can form a weak hydrogen bond, but it's weak individually, but if you spread that out over a lot of molecules, it makes it very stable. This nitrogen here is electronegative, where this hydrogen is electropositive, so they can form hydrogen bonds. This oxygen is electronegative, this hydrogen is electropositive, so they'll interact, and so on and so forth. So water can also dissolve a lot of polar compounds, and since a lot of biomolecules are polar compounds, it makes sense that biochemistry occurs readily in an aqueous environment. So this polarity of water also results in the dissolving of charged compounds. So of course, if you take something like sodium chloride, we already talked about this. So the chloride is is actually fully negative, and the sodium is fully positive. Well, that's the reason why if you mix salt in with water, those salt crystals are going to dissolve, and it's basically going to you're not going to be able to see those salt crystals anymore. And that's because The electronegative oxygens on water are able to interact with the positive charge on sodium, and the electropositive hydrogens on water are going to interact with the negatively charged chloride. So that's why, of course, our oceans are made of many different types of salts, because water is able to dissolve these different salts so easily. Now, just as important as the fact that water can dissolve polar compounds and charged compounds, water does not dissolve nonpolar compounds. So if a compound is nonpolar, it's not going to dissolve readily in water because it's got nothing for those positive charges on the hydrogens and the negative charges on the oxygens to interact with. So if we take this molecule benzene, which we just talked about, so benzene is just made up of carbon with 6 hydrogens on it. Now, carbon and hydrogen basically share the electrons equally. So hydrogen is not hogging those electrons and the carbon is not hogging those electrons. They basically both have the same preference for those electrons. So this is said to be nonpolar. So the same thing with a lot of like lipid molecules. If you think of the fats in your salad dressing or your olive oil or something, those don't dissolve in water because they're just made of hydrocarbon chains, basically. Hydrocarbon chains like this, where it's just carbons bonded to hydrogen. So those have no polarity, so they don't interact with water. However, when these types of nonpolar molecules are exposed to water, they tend to interact with each other. So if you have a nonpolar molecule here and a nonpolar molecule here, they'll actually interact with each other when they're in an aqueous environment. And this actually has to do with physics and entropy and stuff like that, but it's not super important. It's basically, it increases the amount of disorder. So these water molecules want to interact with other molecules rather than organizing themselves around these nonpolar molecules. Not super important. What is super important is that this process is what drives the membrane formation in all of your cells. So cells exist in an aqueous environment. And so the polar part of these lipid molecules are gonna, which are kind of shown right here in the head, those are gonna interact with the water on the outside of the cell and the inside of the cell. And these hydrocarbon chains, so these hydrophobic tails, which are shown like right here, those are gonna interact with each other. And that's going to spontaneously form this cell membrane around these cells. So the fact that water does not dissolve these nonpolar compounds is also really important to life. Your book also mentions that water dissolves gases such as oxygen. That's sort of a more minor feature of water, but it is also really important. So water readily dissolves gases such as molecular oxygen. And also, it's really important that gases are actually more soluble at lower temperatures. So oxygen concentration will increase at lower ocean depths. And this is really essential to us understanding the climate record of the planet. And water also, I think the last thing that I'll mention is that it's extremely useful because it has a really high heat capacity. So those hydrogen bonds between the different water molecules, those bonds are pretty hard to break. And that's why it takes a lot of heat to raise the temperature of a volume of water compared to like the same volume of air. That's why it takes so long and requires so much energy to boil water. And it's also why things like nuclear power plants are always built right next to a body of water so that the water can actually dissolve the extra heat that's used during that's generated by the nuclear power. So this is also really important for regulating your body temperature during temperature fluctuations. So the fact that, it's sort of warm during the day, but if you have to go, if you fall asleep and then the temperature drops overnight, you're going to be okay because the water in your body has a very high heat capacity. So another important feature of water is pH. So pH is it's a measurement of the amount of protons that are in a solution or an aqueous solution, a solution containing water. So in just normal water, a small fraction of the water molecules, which is about only 10 to the negative 7 molar or 1/20 millionth of all the water molecules, this basically is going to occur. So you have a water molecule here, and then every so often, or at a very low concentration, one of these hydrogens will basically be lost and be converted into just a hydrogen without any electrons around it. So it'll be positively charged. And this will leave a hydroxyl group or a hydroxide ion that has too many electrons basically around that oxygen. So it's negatively charged. So this can be written as H2O, breaks down into a proton and a hydroxide ion. So this is positively charged. And this is reversible. So this will go back and forth basically. So the pH, or the acidity or basicity of a solution, is going to be a measurement of the concentration of how many of these protons are in the solution. So pH is just a logarithmic measurement. You don't need to understand that very much. It just means it goes by tens of how many protons are in a solution. So we said that we measure concentration in molarity. So at pH 0, basically 1 molar of the molecules exist as protons. So there's 1 molar, so there's 6.02 times 10 to the 23 per liter of protons in that solution. So most of life occurs around pH 7, which is that 10 to the 7th or 1/20,000th of the water molecules actually dissociate, and there's a free proton moving around in solution. So the pH scale actually measures the concentration of protons in these different solutions. So different biological solutions or fluids contain different pHs. So we usually talk in terms of like physiological pH, and that's around pH 7, which we consider neutral. So human blood plasma is just slightly above pH 7. So that is very important, because if there's too many protons or your blood is too acidic or too basic, that would cause all sorts of problems in your body. So if you think just sodium hydroxide, that would be a very basic solution. There'd be almost no protons floating around in that solution. Ammonia, that is also very basic. Human pancreatic juice is slightly more basic than blood plasma. Something like cow's milk is slightly acidic, so it has slightly more protons than human blood plasma. And then things like tomato juice are even more acidic. And then your human gastric contents, so in your stomach, basically that is a very acidic environment. And that's because your body is trying to break down the food and the proteins and the fats and the carbohydrates that you ingest. And so there's a lot of acid in a solution that helps break down those materials better. So the pH scale actually measures how many protons that are in a solution. So low pH, there's a high proton concentration, and at a high pH, there's a low proton concentration, and that would be considered basic. And so this comes into play when we're talking about acids and bases. So some molecules are acidic and some molecules are basic. So basically some molecules will tend to lose their proton and put the proton into the solution, thus increasing the acidity. And some molecules will accept protons from the solution, which would take protons out of the solution, making it more basic. So an acid is a molecule that tends to ionize to form what's called a proton and a conjugate base. So we have this molecule here, acetic acid, that is a component of vinegar, and it can actually lose this proton here when it's put into solution, and then this proton will just be around in that solution, decreasing the pH. And then it's left with this conjugate base, acetate, that can actually accept a proton, so it's considered basic. So different molecules have a different tendency to lose those protons. So something like hydrochloric acid, that loses this proton very easily. It's very happy to exist in this state with the proton being lost and just the chloride being present in this state. Sodium hydroxide is a very powerful base, so it can lose the sodium group, and then this hydroxide group can actually accept, it's a conjugate base, so it can accept a proton very readily. So if you take an equal amount of hydrochloric acid and sodium hydroxide and put them in a solution, they're basically just going to form water and sodium chloride. So you're going to have, it's going to equal out basically in your solution, and you're just going to have salty water. So, and then the opposite would be when a base accepts a proton. So if we had this acetate ion right here, and then it could accept a base and become acetic acid. So acetic acid, the chemical structure is actually like this. So it's C and then COO and then OH. And so this was that carboxylic acid group that we talked about that's a part of an amino acids. So they're actually able to lose or gain this a proton at this position, which is what makes them acids. And so buffers are very important solutions in biochemistry. So these are solutions that basically you can either make or that are just present in your body that contain both a weak acid, so that's an acid that doesn't lose its proton very easily, mixed with a conjugate base. So this basically buffers the ability for the pH to change in that solution because you have a weak acid. So if you add a base to the solution, that weak acid can just donate its protons to it. And you also have a conjugate base. So if you increase the amount of protons, that conjugate base can accept those extra protons and counteract those changes in acid and bases. So it's not a super important process, but it actually is really important for biochemistry, but not so much for this course. But basically, if you have a solution with a good base in it, with a powerful buffer in it, once you start adding hydroxide ions or protons, the buffer changes the pH from changing very much. You have to sort of hit a wall where it'll start changing a lot. So this is more important to understand just in terms of your blood plasma. So how does your blood plasma stay around pH 7? Why don't you get, these big increases in proton concentration or decreases in proton concentration? And this happens because our blood basically has a buffering system or a buffering capacity. So what happens is the CO2 that we produce by our respiration, that you breathe out CO2, this can react spontaneously with water to form this molecule carbonic acid. And so this can actually lose one of these protons and be an acid. But that carbonic acid can also can actually ionize to form this conjugate base. So your body has this mixture of bicarbonate and carbonic acid that can actually, so if there's not enough protons, this molecule can lose protons. If there's too many protons in your blood, this molecule can accept those protons in your blood. So this is how your blood plasma is able to stay relatively stable around pH 7. Another important feature when you're thinking about water and living systems is osmolarity. So this is the proper balance of charged atoms such as protons, sodium, potassium, calcium, phosphate, carbonic or bicarbonate, and chloride, which is necessary to maintain proper body fluids and pH. And so this is actually regulated by the kidneys. So if you take in too many salts, basically your kidneys have ways to excrete them. And this is because imbalance of these types of electrolytes will definitely affect your cell volume and your cell function. So if you have a cell right here, it has a certain concentration of these ions within the cell. So if you take that cell and put it in a low salt environment, it's going to want to balance out because these ions can move across this cellular membrane. So in a low salt environment, the cell will actually get bigger and expand in terms of volume. And cells can sort of accommodate this to a certain extent, but if the balance gets too big, these cells will basically rupture and lyse, and then they'll basically die, and all the internal contents will go outside of the cell. Same within a high salt concentration. In that case, the cells will actually lose water to balance out water will leave the cell to balance out with the environment, so they'll shrink. Cells are more easily able to shrink in a high salt environment, and this has actually less chance of the cell dying. But osmolarity, just as a general feature, is quite important to biochemistry as well. So, of course, since water is so important to life, that the formation of oceans on the Earth is really an important feature of our planet and what makes it habitable to all sorts of different life. So all the atoms necessary for life were present when the planet formed, but since life is generally thought to have arisen in the oceans, we see how important water is to living systems. The formation of the oceans is basically the catalyst for life as we know it. So the formation of these oceans is believed to be basically from a combination of steam, so water was already present on the Earth, but it was sort of deep below it. So when the volcanoes erupted, this resulted in steam that then landed on the Earth's surface as rain. And this is from these volcanic eruptions. And also, but that only accounts for about 50% of the water. So the other amount of water on the Earth is believed to be from bombardment from comets. So comets can contain a huge amount of water. And over the Earth's history, it was probably bombarded with many different comets. So this was the catalyst or the beginning of the formation of oceans on Earth and therefore life on Earth. And so an interesting correlation is actually the atomic composition of seawater versus living systems. So the salt and the atomic composition of seawater is quite similar in a lot of respects to living systems, although there are some differences. So here's a table here showing the abundance from your book, the abundance of biochemically important elements in seawater and in just human blood in our bodies. And so hydrogen concentration is actually quite similar between the two. Same with oxygen concentration, quite similar. Sodium and chloride. So the amount of salt, sodium chloride salt in the oceans compared to our blood is actually quite similar, which is very interesting. But other atoms kind of differ between the two, including carbon, nitrogen, phosphorus, and calcium. You can see that there's different concentrations of each in seawater versus living systems. And so a big question that people have been thinking about for many, many years is basically the origin of life. So how did life arise on the planet? There's a lot of different hypotheses out there. It just started from small molecules that occurred on the Earth, and then those basically got more complex and turned into life. Some people believe in the panspermia idea, basically where the molecules important for life may have sort of rode on like a comet or a meteor or something and came to the Earth in that way. The answer to this is obviously not conclusively determined, but a really important clue came from this sort of famous experiment in 1952 done by the graduate student Stanley Miller under the guidance of his PI, Harold Urey. So they made an experiment basically where they replicated what were believed to be the conditions of Earth at its early history. So certain molecules that were probably definitely present in the Earth at its earliest time, so methane, water, hydrogen gas, and ammonia, they basically put this into a system, a closed system, and then added sort of sparks to sort of simulate lightning striking and providing energy to sort of make molecular changes. We already talked about how nitrogen gas lightning and UV can change that into ammonia. So they took these molecules and basically added a spark or added some energy input and then and had a heat source right here. It was basically a closed system that sort of filtered through. And what they found was very interesting. They found that they could actually produce many organic molecules that were important for life. So it's not like they got like a DNA or a protein molecule or a lipid molecule or anything like that, but they got simple like amino acids and molecules like that that sort of are the building blocks for these polymers of life. So this experiment has since been modified by other people and it has been replicated. They basically found the same events. So this does show pretty clearly and conclusively that these organic compounds that we've been talking about could have arisen just by the basic processes of Earth and the conditions of Earth in our early history. And so expanding that on that idea, in the Miller-Urey experiments, I mean, they got amino acids and different molecules that are important to life, but they did not get stuff like DNA. They didn't get cells. They didn't get stuff like that. So how did more complex cells and catalytic systems occur on Earth, basically leading to life as we know it? So there's a lot of different hypotheses on how this could have happened, but it's not conclusively determined whatsoever. But basically, the general idea is that once you get these simple prebiotic molecules, that these could basically be over time and with enough interactions and under certain conditions, such as in sea vents at the bottom of the ocean, this could provide the energy and the raw materials to get more complex and more complex molecules. So basically, these prebiotic molecules, so these amino acids or nucleic acids and lipid molecules, could basically eventually interact with each other and form polymers and also really important vesicles. So lipid molecules can form membranes. That's really important. So to have life, you basically need these organic molecules interacting within a cell. So this could have led to protocells, where basically you have lipid vesicles that contain some parts of life, and then that eventually will result in a greater diversity of life. Now, there's a few different things that are quite interesting about this. So we talked a little bit in the first lecture about how DNA, about the central dogma. So DNA goes to RNA and then goes to proteins. So the DNA, the information in DNA basically directs how proteins are functioned. But to get a protein, to have this happen, you basically need the catalytic activity of proteins. And to get proteins, you need the information in DNA. So how do you basically, marry those two ideas? If you don't have the catalytic activity of proteins, you can't make DNA. And if you don't have DNA, you can't make proteins. Well, a lot of people think that RNA is the answer to this activity, and a hypothesis called the RNA world. Now, RNA is a little bit different from DNA and proteins, but it has components of both. So RNA has genetic information, but it also has catalytic activity. So the thought is that in Earth's earliest environment, that RNA, if there was a molecule that was able to both store information and catalyze chemical reactions, RNA is the most likely culprit. So it's a pretty interesting idea. We might talk about it a little bit later. The other situation that provides some sort of evidence toward this is things like the citric acid cycle. So the citric acid cycle, which we'll talk about later when we talk about nutrition, at the very least, is a cycle basically where energy is transformed. So you get glucose in your body. Glucose is transformed to another element, which is another chemical, which is transformed into another chemical that then enters the citric acid cycle. And the citric acid cycle can provide energy from this molecule, and it works in a cycle. So it's sort of recycled at the end of it. Now, this is quite interesting because a lot of people think this citric acid cycle probably predates cells, in that this chemical transformation could have occurred before there was actually living systems, just the cycling of these different chemicals. So that's also pretty important, pretty interesting. So if you had processes that generate energy, like the citric acid cycle, and you combine those in a cellular system and you add RNA, this is sort of the best idea for the origin of life. Now, there's a lot of unanswered questions relating to this, but it is an active form of research. So we'll continue with the next lecture where we talk about these individual building blocks like this, and then we'll continue on to how these form polymers and eventually cells over the next few lectures. All right, so that was lecture number 2, atoms, molecules, and life. Again, I know this can be very confusing. For most of you, this is probably the first time that you've ever played around with chemical structures or thought about how atoms interact to form molecules. But don't get too worked up about it. Don't worry about having to do it on a test or anything like that. It's just interesting information, and it'll help you. The more that you understand it now, the more you're going to get out of the course and be able to, look at the world around you and understand how chemicals work. So I do hope you have a better understanding of molecules and how different atoms interact, because we're going to be looking at a lot of different chemical structures throughout the course. And the better that you understand that, the better you're going to understand when we look at chemical compositions and molecules later in the course. I hope you also understand how important water is to life. So the polarity of water is very important and has some other characteristics that are really important for biochemistry and life in general. And I hope you found it interesting at the end, where I talked about a little bit of the hypotheses surrounding how life basically arose and evolved on the planet Earth. So I hope you enjoyed the lecture, and please always email me or let me know if you have any questions.